Okay, this is a lecture just on the genetics of cystic fibrosis. And I have a couple basic lectures. Some of these were very similar to what you had in the pre-matriculation material, the Mendelian inheritance, as well as tools of molecular genetics. If you have any issues with this lecture, you might want to look at those first just to have a better base. But anyway, cystic fibrosis is a common disorder. 1 in 3,500 individuals of a Caucasian descent have cystic fibrosis, and 1 in 25 individuals of Caucasian descent are a carrier. Don't memorize these exact numbers because you might read a book and one says it's 124, one says it's 126, or some other physician puts in that number. So those are not the important numbers, but they're big numbers, okay? So if you get 25 people in a room, one is probably going to be a carrier for cystic fibrosis. This is much different in different ethnic groups. For instance, in the Asian American population, the risk of cystic fibrosis is 1 in 31,000, almost 10 times less common. So cystic fibrosis, or CF, is inherited in an autosomal recessive fashion. That means both parents have to be a carrier before you have an affected child. When a child is born with a recessive disorder, whether more common like CF or even rare, often you do not see any family history of it when looking at 3 generations. There may be many carriers, but this is the only person that's actually affected. So the molecular diagnosis, there are over 1,300 CFTR gene mutations. A lot in summer when I talk about sickle cell anemia, again, if my life was so easy with sickle cell anemia, there's one change to the gene. There are over 1,300 and counting changes in the CFTR gene, which can result in it not functioning correctly. There are 23 common mutations that represent 88% of the Caucasians, 69% of the individuals affected in the African American population, and 57% of affected individuals of the Hispanic population. So you can see there's 1,300, but there's 23 that are super common and primarily, pick up the vast majority of the Caucasians, but don't really pick up a lot of the Hispanics. Carrier screening is an inexpensive method to look like when, I open up a room, 1 in 25 could be a carrier. Who is a carrier? Our goal in carrier screening is to find people before they have a child that's affected. Sanger sequencing is currently a method used to sequence a CFTR gene, and this is an expensive method, so please refer back to the tools of molecular testing for that. But one thing I want to talk about is the allele-specific oligonucleotide, or ASO, and this is actually a relatively inexpensive method that people use to sequence common mutations. So we're just going to show you the one for the most common mutation, the delta F508. So let's look. So what we do is we have primers for normal hemoglobin, and we have primers for the delta F508 mutation. So if you have normal not hemoglobin, CFTR mutation. So normal CFTR mutation and the abnormal CFTR mutation. So if you have the normal CFTR gene sequence, you will amplify using the primer's DNA. If you have this deletion, and so let's look at this deletion, delta F508. So there's a deletion at site 508 that's 3 base pairs. So instead of it going C, A, C, C, A, G, A, T, it goes C, A, C, C, A, and then it skips this and goes to T, G, A. So we end up with a change in the base sequence, which changes the amino acids that go into the protein. And so we have a primer that looks at the normal hemoglobin and a primer that looks at the I keep saying hemoglobin, I'm sorry, the normal CFTR gene and the sequence for the delta F508 in the CFTR gene. So if we have normal CFTR sequence, we will amplify DNA here. If we have the delta F508 deletion, the primer that's used for that, if we amplify for both, then we are a carrier. This individual also amplifies for both.
They had 3 children. This child only amplifies for the allele, for the delta F508 deletion. This is someone that's affected with cystic fibrosis. This individual only amplifies for normal CFTR, and this individual amplifies for both, just like the parents. So this is known as allele-specific oligonucleotide, or ASO. It uses PCR for amplification and common primer. So this is, again, an inexpensive carrier method. So let's look at carrier testing. And again, I told you there's like 23 alleles that pick up 88% of the Caucasians. And using these methods, you don't need a lot of DNA, and you can do it relatively inexpensively. So let's just look at this. The detection rate is lower in some populations than it is in others, as I said in the first slide. So if you're African American and the detection, looking at common mutations, is 65%, then your carrier frequency before you have the test is 1 in 61, but afterwards it drops to 1 in 75. However, if you're Caucasian and you pick up 90%, even though your risk was 3 times higher before, it actually is lower, 1 in 227 afterwards. Individuals of Ashkenazi Jewish descent, there was actually 3 common CF mutations, and a delta F508 being one of them. And the risk before you took the test was 1 in 24, but the risk after is close to 1 in 400. Asian Americans, their risk is close to 1 in 100 in the general population, but we only pick up 55% with screening for common carriers. So when you're done, you only drop to 1 in 205, and Hispanics, again, 1 in 58, you drop to 1 in 227. So even though they have a risk half that of Caucasians after being tested for the common carrier, it ends up being the same. So what do I mean by this? It's important when you do carrier screening that you know that there is residual risk, and that if you're in a population that you don't really pick up much from this carrier screening, you haven't really decreased your residual risk all that much. So let's talk more about cystic fibrosis. In cystic fibrosis, the gene responsible for CF is located on the long arm of chromosome 7. It's called the CFTR gene, and it codes for a chloride channel in the cell. Medical complications of that chloride channel not working correctly, we historically hear about the respiratory issues, viscous, thick mucus that results in pulmonary issues, infections, respiratory distress. However, GI issues, meconium ileus, so a meconium ileus is when the meconium, we'll learn about this more next year, but the meconium, before the baby's born, there's like all these kind of cells that sit in the GI tract, and the first poop is like this thick, gooey thing, but sometimes it gets stuck and causes the inflammation. 15 to 20% of babies that have meconium ileus, it causes an obstruction before they're born, have actually CF. So it's actually a pretty high risk. And this meconium ileus can result in malabsorption and failure to thrive. Pancreatic insufficiency, this thick mucus affects the pancreas. And then males, bilateral absence of the vas deferens. So the vast majority of males with cystic fibrosis are infertile. And in fact, we used to think there was just one CF, and this is what everyone looked like. And we realized, again, when there's 1,300 different changes in the gene, there's a lot of different phenotypes. There are adult men who come in because of infertility and have 2 milder mutations, but they have infertility because they have bilateral absence of the vas. So as I said, the CFTR gene is a chloride channel that does not work correctly to transport chloride. This results in viscous mucus in the lungs and pancreatic problems. Now, a lot of people say that a baby with CF is a salty baby. The old wives' tales, they would taste the baby and it would taste like salt. And the reason is salt follows chloride. So this is just